Hey Keith.
Hey, Kim.
So I thought today would be really great to talk a little bit about animal models. Animal models are so important for research. They have so many advantages over just doing human research, but then at the same time, there's just a lot to think about in terms of like which model is good for what application, and kind of what are potential confounds. So anyway, I just thought this would be such a great topic.
Yeah, I agree. And I know we're both biased here because both of us work with animal models. So everybody keep that in mind while you listen to this lecture.
Well, it's not like we only work with animal models. That's where we are right now. But my lab is thinking very strongly about doing some human research in this upcoming year.
Yeah, that's true. You're not just thinking about it, you're doing it. So
That's the
I
plan.
misspoke. We're going to be a multi sort of model people soon.
Yeah.
Congrats to us.
Oh well, it's just that there are things you can do in animal models and things you can do in humans, and they're just different. So I think maybe you should just tell us about it.
Yeah, that's a perfect segue into my first slide here. So students, you already know why we do focused ultrasound. When we compare to pharmacology on the left here, we have no spatial resolution, and of course it's bathing the entire brain. And direct current stimulation or transcranial magnetic stimulation are still very poor in spatial resolution. So as you know, focused ultrasound allows us to target these tiny regions of the brain. And that sort of opens us up to this huge dilemma. It's not really a problem. It's almost like a good dilemma. And that is that there are so many different brain regions that we can target. And there's so many different ways that we can pulse ultrasound to affect these brain regions. And there are so many behaviors that we can test with all of these things. And wait a minute, there are also different cell types within every single region. So if you sit there and do this math in your head, like Kim, what are your thoughts when I play this slide? You're uncomfortable, nervous about the possibilities?
Yeah, it's two to the end, or maybe it's like four to the end, or n to the end. There's a lot of possibilities.
More than the grains of sand on a beach or stars in the, well, maybe not that many, but you get the idea. So anywho, how do we address this? Here's where our animal model friends come in. So when you're thinking about animal models, you can think about all of the different trade-offs between our smallest little animal models like C. elegans, this is almost a worm, versus the other side of the spectrum, which is the human. And here we're trading off accessibility versus translatability. So what is accessibility? Accessibility is something like, can we change the genetic architecture of these organisms? Is it expensive to do such, or expensive to run a huge trial with a lot of subjects? And the last one is, can they sort of perform a behavior that's easy to assess? And of course, translatability is the question of, if I've answered a question in this small animal model, does it really matter to the human, and is it the same? So Kim and I deal with these trade-offs quite a bit.
Yeah, I don't know if that really captures everything. I mean, perception is something that's so easy for humans to say what they perceive, and you don't really get that from the other animal models. So that's
Mhm.
somehow that part's not really captured here. But
Oh yeah, that's a good point. So accessibility is actually easier for humans here. So they'd actually be somewhere up here. Don't ruin my graph, Kim. I spent so much time making this, but she's making a great point. Anywho, we're going to talk about a bunch of these animal models and how they sort of compare to humans and different experimental paradigms within each one of them. We're going to focus a little bit particularly on the mouse and rat, because Kim and I hold the mouse and rat so dearly within our own work. But of course, there are other animal models that are much simpler, like the fruit fly, a fish, or C. elegans. Okay, so let's start out by kind of discussing why you might use one of these simple models. And I've shown you all this slide before, but for instance, in C. elegans or really small organisms, it's really easy to change their genetics. In fact, you can feed RNAi or RNA interference constructs to C. elegans by simply dumping it in their little food dish, and that'll silence particular genes within their genome. And then you can use ultrasound to ask the animal, did you feel this? Or did this gene knockout stop you from feeling ultrasound? In which case, the gene is involved in ultrasound sensation.
how do you ask
Kim?
a worm? Did you feel this?
Great. That's a great question. It's as if Kim has seen these slides. So one way that you might do that is by having or tracking an animal's motion through space and then having a small ultrasound field. And if the animal sort of bumps into that ultrasound field and changes direction with statistical significance, then you may conclude that the animal has noticed or felt the ultrasound in response. And of course, you have to do this many times because these animals will randomly turn around on their own occasionally. And that's where statistics come in. Here's a video of a Drosophila larvae sort of crawling around in space, just to give you an idea of sort of the simplicity of the assay. All we're doing here is, or all of this group, Testecin and all, we're just thresholding an image and sort of tracking the position of this animal's nose over time. And then you can say things like, did the animal turn? Did the animal cross an ultrasound field? So on and so forth. And Drosophila has a bunch of its own tools that we can use for editing the genome. One of them is called the Gao4 UAS system. So we talked about this last class. In this case, you have a promoter that is genetically encoded into one sex. Here it's encoded into the male. And in the female, you have a promoter element that is not normally found in the animal called a UAS. And then you would genetically insert some gene of interest. So by crossing these two animals together, you can artificially drive gene X, which might be an ultrasound sensitive gene, in any tissue where the UAS is expressed. So that's a tool that's very easy to use for Drosophila. Here you can see using this system that you can genetically label specific cell types in the brain or the ventral nervous or the ventral nerve cord, shown by these two different colors here. And you can actually differentiate them by using series of different promoter systems.
Are people using Drosophila for studying ultrasound?
Not that I know of. I actually put this in here almost to encourage people in Drosophila to get into it because it's so similar to C. elegans. And you know, I mean, who do you know that uses C. elegans a lot?
You know, Jan Kubenek did some work with Miriam Goodman.
Yep. So even, you know, two people that Kim is very close with. I know Jan used to be at Stanford here. They use C. elegans in really awesome ways that we might show later. So I'm hoping people adopt it in Drosophila. I used to work in Drosophila. That's why I really love them. Great model organism.
Well, you know, the worm is just easy to couple ultrasound because they're like in a water bath or something. But with the fly, it's in air. So coupling maybe is an issue.
Oh, so I should say that when a Drosophila is a larvae, so this is actually a larvae over here, you can put them in a fluid solution or you can put them on top of an agar plate. So you're actually getting direct coupling. And there are probably more labs using fruit flies than there are C. elegans. So that's why I kind of want people to get into it. And but like you said, it hasn't picked up yet.
And do they mostly use it in the larvae state or once it's a fly and flying around?
It depends on the behavior. That's a great question. So if the larvae does something that you're interested in, you definitely want to go with the larvae because you don't have to keep them around and alive long enough. But I used to work on sleep. And even though larvae do show some phenotypes of sleep, the adult fruit fly was much better sort of model for sleep because you can measure optical activity in the brain. You can watch them walking around. You can do EEG, all that stuff, but you can't do it in the larvae per se. So this is an organism that neither Kim or I have experience with, as far as I know. This is a larvae zebrafish. And here you can see that you can actually track the motion of the larvae very easily in solution. And as we know, having a water tank allows for very easy ultrasound coupling. Now, the really cool thing about the larvae is that you can actually head fix these guys with a little bit of sort of crazy glue. And you can use a little bit of anesthesia and actually measure what you think they want to do using electrophysiological pins in sort of their muscles. So here you can see an animal that's head fixed and the system is sort of assessing, does the animal want to turn right? Does the animal want to turn left? And at the same time, because the animal's cuticle or sort of skin, I guess we wouldn't call it cuticle, but let's call it skin, is totally transparent. You can measure activity from every neuron in the entire brain simultaneously. And this was using light sheet microscopy by Dunn et al. So anyway, I think there's a little bit of stuff in zebrafish, but there should be more in my opinion. Thoughts, Kim?
I think there was some study by the University of Utah looking at vestibular effect in fish. The vestibular effect, like
What's
how
the
does
vestibular
ultrasound?
effect?
Well, how does the
Or
ultrasound affect the vestibular system? And they found that it did.
what is the vestibular system?
Well, so it's part of the inner ear, and it doesn't tell you about hearing, but it's near the cochlea, and it tells the animal whether they're accelerating in one position or, in one direction or another, whether there's circular acceleration or linear acceleration.
That's super interesting. And did they find any modulation of that system?
Well, they did find that ultrasound affected the vestibular system. And if you think about how ultrasound might go through all the soft tissues, and then maybe it, because the way they work is that there's sort of loops of fluid, and then there's otoliths in there, which are like little calcified stones, and they move back and forth. And I think they have hair cells as well, and that they can detect the motion of those little stones, and that tells them about that the acceleration. But the key is if you have ultrasound sort of moving the fluid or maybe moving the otoliths just by ultrasound itself, then maybe it's picked up as like, oh, I feel like I'm accelerating.
Wow, interesting. It sounds like a target that would be really sensitive to ultrasound because you literally have something moving in fluid, which we know we can do very well with acoustic streaming. That's super interesting. Kim, you're gonna have to link that for the group later. Okay, so moving on to mice. Just to show you guys, it's also very easy to track a mouse. So here I wrote a Python script that's just outlining sort of the black contrasted against the white background. And you can do interesting things like splitting the arena. Here I have a plastic piece of glass on the right and normal bedding on the left. And if we stimulate the animal, you can see the blue light indicates stimulation when they cross over. If we stimulate them only on one side, we can start to say, do the animals like the ultrasound by asking whether or not the bias for the plastic or the bedding shifts when you deliver ultrasound. So fun experiments like that are things that Kim and I are super keen on with the mouse.
Well, what did you find? Does he like the ultrasound?
Oh, I actually didn't end up pursuing this particular experiment just yet. I focused more on epilepsy. I do think Jurhai has been using my chamber for this though. So we'll have to ask him if the results have been good. Okay, so let's move on to different neural activity readouts. So you got a sense of easy tracking behaviors. Of course, there are many more. You can look at individual limbs. You can look at preferences for taste and things like that. But what if you want to record the neurons directly so that you're really asking right from the source, what's going on when I deliver ultrasound here? So electrophysiology methods are sort of the standard. They've been around forever. These things are the crux of modern neuroscience. And I just wanted to show you sort of the basic idea here. And here's a simple circuit diagram. And I'm just trying to, I know you have enough experience at this point with the course, but the idea is that you have the outside of the cell and that the membrane acts as a sort of resistor for current flow, or even a capacitor in many ways. And then you have the inside of the cell, and that when there is an action potential and ions cross this resistor, you get a change in voltage, which you want to measure and indicate that a neuron is doing something. So probably the easiest method, probably some people would say, oh, it's not the easiest, but I would say the easiest method is something like taking a sharp electrode, and these are typically long glass tubes that are stretched out really quick and cut so that you get these really sharp ends, and you'll fill them with solution and put a metal wire in them, kind of like you're looking at here. And then you can actually poke them to the inside of the cell. So now you have access to sort of the ionic content of the inside. And on the other side of the cell, you just stick the electrode, sort of the ground or reference, anywhere outside of the cell. Just has to be nearby. And now you're measuring the voltage difference between these two states. You can also do things like asking whether or not stimulation of a presynaptic cell, So here we have a presynaptic neuron in red that when we stimulate should sort of dump these neurotransmitters to our synaptic boutons or on this other cell, this postsynaptic cell. And then you can measure whether or not this cell is responding to that. So you're looking at circuit strength. And here's a sample of what that recording might look like. Here's the stimulation artifact because we're driving a strong sort of electrical signal here. And then here's the amplitude of our postsynaptic cell over time. And you can do simple things like delivering ultrasound and then asking if our response amplitude drops, where it might look like this, or whether it increases and looks like this. So you're asking, is the cell more or less responsive to an external stimuli? And then on the right-hand side here is a patch clamp diagram. So patch clamp, I think Merritt probably already gave you a brief on, and I might have as well. But in this case, you have a sharp electrode, or an even more sharp glass electrode, I should say. In fact, these are really only on the order of a few microns wide when you get down to it. And you'll actually smooth out the end with like sort of a blowtorch almost, like a lot of heat. So these are very smooth, but still very fine. And you'll actually poke it just to the edge of the membrane under a microscope. And then if you apply a little bit of a negative pressure, you actually connect your electrode with the membrane of the cell. So now you have access to sort of the individual ion flow and high resolution. And there are other variants of this where you can pull pieces of the membrane off so that you can look at individual channels rather than the whole membrane. And this goes on. I should point out that this method is very difficult with ultrasound. Kim, can you tell us why? Not to put you on the spot. Do you remember why? I can ask.
Yeah, well, so Merritt had a really nice video. I don't know if you have it to show, but just that when you apply ultrasound, that there's like the cell itself seems to change. And one of the questions is, what about the fact that it's going through high pressure and then low pressure, but yet in the pipette, it's not really changing its pressure. So is the cell kind of going up into the pipette and out of the pipette? And, it was kind of confusing to me.
Mm-hmm. And ultimately, though, you're getting at that the cell is like moving around quite a bit. And this seal between the cell and they often call it a glass pipette, but we'll just call it a patch clamp. This seal is extremely sensitive. So even when you're working with healthy cells and not delivering ultrasound, sometimes it'll just break loose. So you can imagine radiation force moving the cell easily disrupts this and ruins your recording, as Kim was describing. I don't have that video, but we've probably showed it a bunch of times by now. Maybe the students remember. Okay, so we just described a problem that is a really huge problem with patch clamp because it destroys your recording. But what if when the cell was moving, it was sort of ruining your sharp electrode recording, but you didn't know it? And I want to tell you about when that might happen and when it might lead to an artifact that you think is real signal. So Morgan Collins et al. Had a really nice paper in eNeuro, I thought, where they splayed open a leech, which is this animal model that has these really huge cells, these huge neurons sort of right at its core. And these are called Retzia cells. And the reason people love to use them is because they're so easy to see, I think, under a microscope or even free fields, and to poke your sharp electrode inside of them. And what they were doing in this experiment was having a splayed open leech on a glass slide and having coupling to an ultrasound transducer. And while measuring a sharp electrode vertically from the top here, they would deliver ultrasound from the bottom. And the ultrasound peak negative pressure, or the pressure field, is shown here. So you can see they're delivering plenty of pressure, or at least a lot of the field over the splayed out leech here. And here is the interesting finding. So if you look at a baseline electrophysiological recording, here's a scale for 10 millivolts for reference. When a brief ultrasound pulse of 138 kilopascals is delivered here by this little spike, you can see a nice response in electrophysiology. So clearly the neurons are doing something in response to ultrasound. The problem, and sort of the whole crux of this paper, is that when you did sort of a very similar sort of perturbation, but instead of delivering ultrasound, you actually move the pipette up and down slowly, where it still maintains contact, but it's physically perturbed in space, and that's shown by green here. You could get a deflection in the electrophysiological signal that looked very similar in amplitude to our ultrasound perturbation. And that's quantified on the right here. So here you can see that as pressure is increased for the short pulse, you get depolarization that looks about 20 millivolts. And by displacing by some amount of microns, you can get an extremely similar effect. Kim, so I guess here's a question that I have in mind. Is micron displacement on the order of what we would expect with radiation force from your previous lectures?
Yeah, exactly. It's on the order of microns. Yeah.
It is. So, okay. So Kim and I both agree that this is a huge problem potentially if you're recording with your electrophysiological method from the same area that your Retzia cells are, or sorry, your neurons of interest. And I think the idea here is, because we're not going to discuss it within the paper, but if you have a voltage that's very sensitive to fields placement, so these fields are very small, and you move the entire recording electrode, you're moving it across this field. So you may get a deflection that looks as if a voltage change is happening when really just the change is sort of where you're measuring in that voltage field. Does that make sense, Kim? Am I doing that justice?
I think so, but I'm a little confused that aren't you saying that if you move your device by just a little bit that's on the order of the radiation force that you're going to get the same sort of deflection? I mean, isn't this sort of saying that the mechanical force has the same response as the ultrasound?
It is, yeah. You might make the argument that moving the needle itself is affecting the membrane and delivering mechanical force to the membrane. But I guess they were making the point that it wasn't ultrasound per se, but more an artifact. But Kim, you're making a great point here. That's why. But one paradigm that they came up with to ask whether or not ultrasound was having sort of an offline effect, where the glass electrode was no longer present, they deliver ultrasound and then try to record from the cell again. So the idea here is that you place the electrode in cell A, this Retzia cell, you remove it and deliver ultrasound. So there's no sort of membrane electrode interaction, as Kim is suggesting. And then you place the electrode back in and record the results again. Now, this poses a little bit of a problem because the actual injection and removing of this electrode is going to mechanically perturb the cell, kind of like ultrasound would do, right? As Kim already mentioned. But the really nice thing about this system is that there's actual connectivity between other Retzia cells in the neighboring area. So here we can actually flip over and record from another Retzia cell that has not been exposed, and that will be a proxy for our connected neighbor, if that makes sense, which was not damaged previously. Okay, and here's what they found. So here is sort of a demonstration of what the ultrasound looks like. We have membrane potential on the y-axis and sort of the pre-ultrasound signal across time. So you can see there's sort of sporadic spiking of these neurons in response to stimulation. And then here you can see post-ultrasound response, where the membrane potential is certainly different. And that's why it's important to measure from the contralateral cell afterwards. So as you'd expect, under any conditions, I suppose, if you believe in ultrasound or mechanically disrupting the cell, you can see that in our same cell recording, the control had some given resting membrane potential. And then relatively after ultrasound exposure, when you recorded from that same cell, the membrane potential has significantly changed. What was interesting here is that the control in the contralateral cell, when you did the control measurement in cell A, and then you recorded in cell B following ultrasound exposure, there was still a change in this cell and what it did. So I think the group was trying to make the point that there actually wasn't. Here you can see there's sort of a non-significant here, and that they were trying to say that ultrasound didn't matter, and that it was actual mechanical perturbation of this cell that caused this difference. But in my mind, there's definitely a difference here, and that this sort of one sporadic point has lost significance. So anyway, open to interpretation, and you should definitely go and read this paper. But I think it's important to note that mechanical deformation, as shown here, that is not ultrasound, can also change neuronal activity, and that it needs to be considered. Kim, maybe you can give the wrap-up statement here if I didn't do it justice.
No, you do it.
That was it. That was the wrap-up statement. You got to consider it. Just make sure when you do your work, if you can ideally record outside of your field, your modulation response, or offline, or use a non-invasive method, which we're going to get into, that would be ideal.
Yeah,
okay,
we'll
so
talk
just
about that later. This is one of the reasons why I really like the offline measurements when we get to Leonard Verhagen's talk, for example.
yeah, that's right. Leonard does a lot of fMRI post sort of ultrasound stimulation, and Kim and I agree that's a really good way to do things. So let me talk a little bit about local field potential measurements. So these are really the simplest thing you can do for recording from the brain. I routinely do them for epilepsy research with the Saltez lab and Jordan Farrell. In this case, you would have a bundle of individual little electrodes, in which case they would serve as grounds and leads for one another. And here you can see that all of these electrodes in this drawing have different lengths. So you're essentially measuring changes in potential across a field of many neurons. So if there are neurons active that are not perfectly synchronized, you'll start to get voltage changes, which indicate neural activity. And as we already mentioned, it's best if the electrode is not underneath the ultrasound, because here you can imagine all the neurons being pushed away from the electrode would give you a huge change in electrical voltage, and it would look like activity. Or an alternative is offline, as Kim mentioned. Here I just wanted to give you a flavor of what a deflection, or what you can record from these. So here I'm detecting seizure events with a two-prong electrode or a bipolar electrode. And that signal would look like a very large amplitude slow rise. They call these high, and then you have these sort of high frequency or HFO oscillations that ride on top of this large amplitude event. So in your electrical signaling, you can look for different frequencies, and you can filter and find all kinds of interesting things happening in the brain. Again, here's a seizure in the brain. And here I'm plotting all different seizure events relative to ultrasound stimulation at time 0. And you can see that seizure event amplitude and probability of happening, how many dots are there, goes down substantially with ultrasound, and that it has a little bit of a lag effect. So there's some offline effect here.
Wait, is this data that you measured?
Yes.
Oh, I'm not sure I've seen this data. This is really interesting.
Kim has seen it, so she did see my talk at the Focused Ultrasound Foundation, but it's always good to see things twice. So yes, so Kim, this is some new and fresh, hot off the press data.
Hey, thanks for calling me out on that one.
Maybe she was kidding. She knew she saw it, but keep things interesting, Kim, your favorite method of monitoring neural activity? Using fluorophores. So what is a fluorophore? And again, we've already told the class a little bit about this, but here's a diagram. So a fluorophore is something that absorbs and emits photon energy at different wavelengths, or they can be very similar wavelengths, but ultimately different. So you can imagine you have some molecule with some valence shells, and you pass energy with light into this sort of molecule or protein. And this energy leads your electrons in these valence shells to jump to a higher state temporarily. This is typically, in most molecules we're looking at, very quick. So as soon as they're sort of raised to this higher energy state, they quickly jump back to their lower energy state. And when they jump to this lower energy state, that energy has to be conserved, as Einstein once told us about the conservation of energy. And it will leave this system as an emission of light, very similar to how it came in, but a little bit lower energy. So there's some energy lost in this system. maybe as heat, as thermal energy. But the light is emitted at a lower wavelength, and we can actually measure this emission. So in this case, we know there's some absorption and emission. And with our favorite fluorophore, which is GCAMP, this doesn't really stand for anything explicit, so we'll just call it GCAMP. There's a difference in your excitation and your emission spectra. So you'll push in blue light and it will be emitted back as green light. So your detector and your emitter actually have to be sensitive to different wavelengths. And they call this the Stokes shift, is the shift that I described in wavelength between these two peaks. Okay, so let's talk about genetically encoded calcium indicators, which GCAMP falls under that category. You know, what is a genetically encoded calcium indicator? So this is very simply a fluorophore which depends on the presence of calcium. So here's a sort of molecular diagram, a spatial diagram of this particular molecule. And you can see that there's a little binding pocket for calcium. And in the absence of calcium, this large molecule will actually shift relative to our sort of implicit fluorophore, green fluorescent protein. So if it shifts a certain way, it will not fluoresce. If calcium is present, these molecules will bind together and become fluorescent. Okay, so this is, of course, pertinent to electrical activity in the membrane, because when there's resting membrane potential, there is extremely low concentration of calcium inside the neuron, as we already know. In fact, it's as low as 100 nanomolar relative to 1.5 millimolar. That is a huge difference in calcium. However, when the cell becomes active, we remember that calcium channels, which are voltage dependent, open up and allow a flow of calcium into the cell. And this causes a dramatic increase in this concentration, such that these fluorophores now become active. So, okay, so you can imagine if I'm pulsing blue light into the system and I'm monitoring the green light that's coming back, when a cell becomes active, you'll see this large, fast rise in fluorescence returning. And that's telling us that calcium has changed the conformation of this molecule. There are many variants that people have genetically engineered, such as GCAMP slow. So in this case, the kinetics of the binding and unbinding of calcium are very slow over time. So even a single neuron becoming active, you'll have many, many seconds before this calcium gets removed. So the fluorophore is fluorescent for a long time. Then you have other variants that are very fast, such as GCAMP fast.
So this term slow and fast mostly referring to sort of the recovery phase, not the excitation, because they look like they're going up with sort of the same slope there. So they're both responding quickly, but fast and slow means how fast they recover.
Yes, and the word recovery should really be swapped in with unbinding of calcium.
Yeah.
Sort of the recovery of the initial state where there's very little calcium bound here at all. So that's a great point, Kim. So they all bind at the same rate, but this GCAMP slow takes a long time to unbind.
So let's say you're doing an experiment and you wanted to do multiple measurements so that you could build up your statistics. If you use slow, then that means you have to wait a longer time before you can redo the measurement.
It does. Yeah, that's a great point. So if we're using GCAMP slow, we're not going to be able to resolve different events very easily. Kim, so in that case, you'd probably recommend GCAMP fast. Do you know why offhand we might want to use GCAMP slow, where our temporal resolution is clearly worse?
Well, I mean, it looks like you get more fluorescence change. And so if you're talking about signal to noise, you could go with the slow and get more signal change and do it less often. Or you could go with the fast and you get less, and then you do it more often. And I don't know if it makes a difference. You know, it kind of depends on the exact numbers, and maybe it all comes out in the wash.
No, Kim, you said it right. It's better signal to noise ratio. You have longer accumulation times. You have more cells that are holding on to calcium at the same time. So you're getting a larger response. So the trade-off is temporal resolution versus SNR. And I got to tell you, everybody, these genetically encoded indicators, they're very finicky. Whether or not your virus transduces the cells very well, it can be difficult to get good labeling sometimes. So that can be quite important. Just to give you a working knowledge of like a way to actually quantify your effect. And everybody has their own flavor of doing this. You know, courses are taught on different methods. You just have to read between the lines and ask how each group does it. And the running idea is that you have some sort of a baseline period. And you know, during your baseline period, the neurons can be active. They're going to look like whatever the animal is doing at that time. And the idea, though, is to provide some sort of stimulus and to quantify how much your effect period is changing relative to the baseline. Sorry, the baseline. And the very typical formula and notation for this is delta F over F, where the F over here refers to the baseline and the F over here, or the total F, refers to the change relative to the effect. So here you would subtract the difference, normalize them to baseline, and people typically multiply by 100. You don't have to do that. It's arbitrary, but you can. So whenever you look at fluorescence data and relative values, just know that they don't mean anything at all without a control. So you can like do crazy things to these numbers, and someone might say, oh, they have a delta F over F of like 100. That's really big. It's not. They're just making up different ways of analyzing. You always have to look relative to the control.
And so the signal can go down as well. So you could have a negative delta F over F.
Absolutely, you can. In fact, if you're looking for inhibition, you really prefer a drop in effect. Mm-hmm. And Kim already mentioned this, you know, why use GCAM slow? It's because you can get an aggregate. So a single neuron might have a profile that looks like this nice smooth curve, but if you start adding the fluorescence together, you get sort of a noisier, bumpier, but ultimately larger aggregate signal. And just to let you know, this is what your signal is really doing. It's integrating or sort of adding together all of the individual cells in the field of view that are outputting signal. Okay, so this is Jamie Tyler's sort of one of his earlier sort of revival experiments where he was imaging fluorophores in a dish of the CA1 of the hippocampus sliced out of an animal. And here you can see all these little neurons sort of lighting up. And when the stim comes on, you can see many more neurons becoming bright. This is giving you an idea of what the actual data looks like so that you can grab onto it with your mind. And I use his data to not only show you like how to work with this data, but also, you know, having ultrasound data is kind of exciting to play with. So what I did here was I actually downloaded his video so that I could reanalyze it on my own. And you can feel free to do this sort of thing for your project. It's totally okay to reanalyze people's data. And here's a basic way to do that. So you'll take an image series. You can download it into any software you want. I did ImageJ here, and I thresholded to a binary, I think this was a 16-bit image. So here we have just gray values that are sort of across space. Maybe it was 64, I can't remember exactly. And I actually drew individual circles, or ROIs, this means region of interest, around individual neurons. And the question I wanted to ask as a scientist, and again, anyone can do this, very easy, is do all neurons have the same response profile to ultrasound stimulus? And I didn't even collect this data myself to show you how everybody can go in and be a scientist. Here I've dubbed these cells 1, 2, and 3. They're color-coded here. And here you can clearly see at frame 9, when the ultrasound comes on, a large rise in all of these cells. But indeed, as I hypothesized, it's a fun science term, these cells have different magnitudes of response, and even a couple of them may have sort of this post-ultrasound response, although you may argue statistically that these are sort of stochastic events that are unrelated to the frame when our stimulation comes on. Kim, you want to say something? What are you thinking about this experiment?
No, I think it's just very cool that you did that with that particular data set, because it never occurred to me just to reanalyze it, but that's very cool. But it's very interesting to me that you see these spikes later, but yet you didn't see spikes earlier. And so is the later spike causative from the, caused by the ultrasound or just, yeah. What do you
I
think?
don't know. You'd have to do statistics. Also, Kim, you're saying something really awesome that I'd like to point out. We don't have a very long baseline here because Jamie just didn't give us a long enough video. But if we had drawn this line out very far, we had more video, we might start to see some random sort of spikes. In which case, then we could determine if this spike over here is stochastic or statistically a real effect relative to our ultrasound here. And Kim is a scientist, so I'm glad she brought that up. It's like a team of scientists thinking together here.
It's one of the things when I got into this field that I really had to learn was, in physics, it's like you do something, you expect a result, and it always happens the same way. But when you're talking about biological systems, it's all about the statistics and putting together lots of measurements. And
yeah, that's absolutely right.
Yeah.
It's a crazy field. It's like poorly defined in all means. And I've heard the same thing about physics experiments. They're very clean cut, very reliable. Not biology, everybody, not biology. So let's see, where are we headed with this exciting lecture? Oh, to give you a little bit more calcium imaging sample data. So here is photometry data from my own work. And photometry data is where instead of analyzing those little individual cells, you just have an optical fiber that collects all the light from the field. It's just everything is just coming in. So you're looking at all the cells working together, and here's a trace of that over time. And I want you to notice that this actually trends downwards over time. And that's because these molecules, these proteins, will actually bleach. So the bonds, the double bonds between oxygens will actually break from the light energy, and you'll get less signal over time. It's an important trade-off of using optical fluorophores. And here you can clearly see sort of non-active times versus active times during these peaks. And here I mentioned the trade-off is, do you want to use more excitation power to get more light signal back, better signal to noise ratio? You might, but remember that when you increase this SNR, you're also going to get more photo bleaching. So your signal will decay in sort of amount of fluorophores you can measure with over time. Something to think about. And Kim already mentioned, well, one method that we could use is to deliver ultrasound to this system. And over many, many, many events, you can ask, do these statistically seem to respond at the same time across trials? Because again, these neurons may just randomly do things independent of ultrasound. So this is how we would do that. And here I've delivered 20 different trials, and you can see that reliably the neurons respond to ultrasound. And here's an average plotted with standard error of the mean here. Okay, I really quickly wanted to say that there are non-calcium dependent, well, sort of calcium dependent fluorophores called genetically encoded voltage indicators. And these are a little bit different in that they sort of operate on the specific voltage across the membrane. So here we have the outside and the inside of the neuron. And here are two different types, you might say, where some sort of fluorophore may be very sensitive, I think I have, to a negative charge that may be inside the membrane. So Kim, we remember that there's a negative charge inside the membrane during resting membrane potential, but what happens when neurons become active?
Yeah, it goes positive.
It goes positive. So it might create sort of like a torque with this sort of channel, and this conformational change that occurs here may cause this molecule to become fluorescent when it otherwise was not. So here you can see the FlickR1 voltage indicator. And on top, you're looking at fluorescence changes as a neuron is spiking over time. So it's a fast spiking neuron. I don't know which type of neuron exactly. And at the same time, they recorded the millivoltage with patch clamp in this neuron or sharp electrophysiology. And you can see that the signals are very reliable and highly correlated. So like you might look at this and be like, that's awesome. Why are we not all using voltage indicators all the time? And the reason for that is that the signal to noise ratio is awful for these things. So like this is your very best case scenario, this plot I'm showing you here. But in practice, these things are really hard to get good signal out of compared to baseline fluorescence that you'll get. They are, however, sensitive to membrane potential, whereas genetically encoded calcium indicators are not necessarily responsive to small changes. So let's say the membrane becomes slightly depolarized. Let's say you provide ultrasound just a little bit and you're shifting sort of the resting membrane potential from -70 to -65. So this change is not enough to ungate voltage-gated calcium channels, but it is relevant because it makes our cell more excitable. So only our voltage indicators might actually detect that. The other trade-off is that it is sensitive enough to single spike detection. Now I put the maybe in response to genetically encoded calcium indicators because some people argue that GCAM-FAST can detect individual spikes from neurons. But that probably depends on how quickly the spikes come in succession, because they have to be small enough so that that sort of trailing edge that Kim mentioned could be a problem. You know, it may be a problem if the spikes are really, really quick. Whereas with these GEVIs, it's definitely not a problem. Okay, so I spent kind of a long time on that slide. My apologies. I just wanted to show you one other fun thing about imaging, and that is that these imaging molecules are all genetically encoded. That means that using genetic tools like drivers and promoters, I can drive certain fluorophores in one cell type and other fluorophores in a whole nother cell type that has different promoters present. So here I'm showing you some of my own data. I think this was dopaminergic versus GABAergic neurons in the brain. And here you can see that when I deliver ultrasound, this is the average across many trials, these red GABAergic neurons are highly responsive and it's almost immediate, whereas these excitatory neurons during that same period don't do anything at all. So it's cool because, okay, I have selectivity with my ultrasound, and I also have this delayed sort of dopaminergic response, which gives us all fun sort of thought experiments. Kim, what thought experiment would you have after seeing data like this?
I'm not sure.
Okay, I'll give you one. That's okay. I'm putting you on the spot very quickly. One question you might have is, does the ultrasound have a delayed response in these dopaminergic neurons? Or are these GABAergic neurons somehow interacting and causing this delayed response? Perhaps their strong activation is inhibiting all of what would have otherwise been ultrasound excitation here. So anyway, there's a lot of fun things you can think about when you start using imaging tools that with two different cell types in the same region. So for the next readout of neural activity, we're going to look at something a little more translatable and a little more whole brain. And what I want to describe is PET imaging. So we've talked a bunch about this. This is positron emission tomography. And the idea here is that you have some sort of radio labeled molecule. In most cases, people use something called 18-fluorodeoxyglucose. So this is a radio labeled glucose molecule that when you inject it in an animal, and I'll just do a little drawing here. Imagine we have the brain. This molecule flows up into the brain through the blood system, and wherever the brain is most active, let's say it's right over here, all of your molecules, or most of your molecules, will be absorbed in the highly active areas because brain regions use glucose for energy sources. So to try to understand where these molecules are localized in the brain, you essentially put someone in a giant detector ring. This is a PET scanner. And as these molecules annihilate, they essentially send out these, I guess they're like photons, right, Kim? Like what's radiate?
Yeah.
So the photons are shooting out and you can detect the time it takes for them to get to, you know, each detector, and you can localize where the source of them are. And you can aggregate that signal over time. And for our experiments, just as an example of how you might use this with ultrasound, I went over to NIDA to work with a few folks where we stimulated the brain, a particular region of the brain, for 30 minutes. And then we popped the animal in a PET scanner and sort of looked at where glucose had been taken up by the cells. And here I just wanted to show a simple example of a brain where I was stimulating this region of the brain here. This is the hippocampus of the mouse. It's involved in memory formation and location tracking and things like that. And here you can see that the ultrasound stimulation has suppressed the uptake, which is color-coded here. So there's sort of less red in this region and in the region right below it, compared to the other hemisphere, which served as a control in this experiment. And in order to quantify the difference, you could do something like flipping the brain around and sort of comparing it to itself. Okay, the last method, which is a little older, so this method's been sort of around, it's sort of a staple of neuroscience. I think it's being used less and less, but this is something called Phos Immunostaining. So Phos is an immediate early gene that's highly expressed following neural activity, as we have written here. So how can that be useful for sort of recording neural activity? Well, the idea is that you have a neuron, this little black drawing here, and let's say the neuron lights up and you wait for this gene to be expressed. So you're waiting 90 to 120 minutes. So in this case, maybe ultrasound was stimulated during this neural activity period, or maybe not. And then essentially you are fixing the brain by perfusing in something like paraformaldehyde or something of the like. And you're pulling out the brain and you're using little antibody ligands for this Phos protein, which is expressed as a result of neural activity. And you can attach a fluorophore, which we've already learned about, to these antibodies. So then you can use an optical readout of where neurons were more or less active, depending on how much Phos is present in the brain. So people still use this. Kim, thoughts?
Yeah, no, I have a question here. So if the CFOS is made in the cell and your antibody that you're labeling it with is something that you're injecting it, how do they meet up?
So I mean, I guess the way this works is you put the brain in a little well, you inject with a pipette your antibody, and you just shake the dish around for many, many hours. And the antibody, it's a large protein, but it's still small enough to sort of make its way into all little pockets of the brain.
Okay, so it'll diffuse in, but this is after you've sliced it. So okay, yeah.
Oh, that's a great point, Kim. Yeah, you have to slice it, or there's no way that antibody is going to get deep in the brain. That's a good point.
Yeah.
A good question you're raising is whether or not Phos presents on the cell surface or not, and I don't know the answer to that. We should look that up. So Kim even still uses this method. I don't use this method, but the reasons that Kim might argue that it's still good is that it's non-invasive, so you don't have to do anything inside the brain for this to happen. It happens all on its own, and it's pretty simple. It's straightforward to do, as I've described here. The problem is, this is the major problem, is that it's really low temporal resolution. So this sort of response is aggregated over a very long period of time, and you need a lot of cell activity to get a nice signal, or else the brain would be just totally full of Phos, because as we know, all neurons in the brain are active at some point across, short periods, right?
And if you're doing this in vivo, then you just get one shot.
Yeah. Oh yeah, I should put that. You have to sacrifice the animal. I mean, you probably got that from this perfusion. They don't survive with one shot.
Well, on the slicing part too.
Oh yeah, that was a dead giveaway. A dead giveaway, because the animals are dead after that. So another thing that a lot of people don't know, Kim, I don't know if you knew this before I had mentioned it, but it's not equally expressed across cell types. So this is a very preferential for excitatory neurons in the brain. Inhibitory neurons don't express that much Phos following activity. So you have to be sort of careful when you interpret your results around that. Okay, so as I keep pointing out Kim, I have some slides from her old paper. Kim, do you remember this sort of ballpark experiment? I only have 2 pictures here, but
yeah, I do.
Please explain.
You want me to explain?
Remember it?
Well, I think it's just kind of straightforward here. There was the control mouse, and then we did some Phos staining, and then the one down below, we did the ultrasound, and then we sacrificed the animal, did the Phos staining. And so you can see that there's more bright spots down below. And then we went through a phase of quantitating that using NIH ImageJ and pulling out the different ways you can do that. You could take like a large region of interest and look at the fluorescence, or you could try to just quantitate number of cells. And ImageJ has capabilities for sort of segmenting out and quantitating cells.
I'm glad you mentioned that, because earlier we showed the students how you can threshold an image. And here you can see that these little, well, I guess my color of my thing is wrong here, but you can see these little speckles that are showing up, and you can threshold until you start to see sort of a cell-like pattern. And then ImageJ can go in and, like Kim mentioned, just count them like this. And you can see that in the control mouse, there's very much less cells labeled here. So Kim's experiment was a phenomenal success.
We never actually, I don't think, published that. I think we talked about it a couple of meetings. It went in Patrick's thesis.
Oh, I wonder how I got it. Maybe you sent it to me.
Yeah.
Anywho. Let's see what I have on the right-hand side here. Oh, okay. Yeah, I wanted to point out why this, another reason that CFOS could be tricky relative to something like GCAMP. Okay, so on the right-hand side here, I have GCAMP recording from the thalamus excitatory neurons. So, tell me what you're seeing here, Kim, across time, if that's all right with you.
Okay, well, let's see. So I'm assuming ultrasound goes on when you have the gray bar.
That's right.
And then you have more fluorescence immediately afterwards in all neurons that you're recording from. Mm-hmm. And then there's a decrease, but the decrease isn't the same amongst the different neurons.
Mm-hmm. So, and this decrease is sort of like a long time, right?
Yeah.
So imagine we're trying to use CFOS to detect this short pulse and whether it activated cells in the brain. You can see that we get an aggregate like fast increase, but this drop off would probably negate, some accumulation of CFOS. So we'd never know that this happened over this short window. In fact, we might even see less CFOS. So we might just interpret it as ultrasound shut off the brain, when in fact we needed this temporal resolution to say that there was sort of a bimodal switch. So just kind of finalizing the argument that CFOS is a good quick proxy for like net activity, but you don't want to use it for fine resolution on a short time scale.
Yeah, but wouldn't that always be true, that you would always have a quick spike and then a decrease after that?
No, not necessarily. So I have some neurons that if you lower the intensity of ultrasound, you'll get a spike and it'll come back to baseline and rest.
Oh, and just baseline rather than.
Yeah, and then there are some neurons that even continue to be active for long after. Okay. They do different things.
Okay.
I actually think that might be. Oh, okay. So here's where we sort of can intro into Kim's really interesting lecture that's coming up. There are many well-characterized behavioral paradigms in mice and rats. We showed some of the tracking earlier, and that's why Kim and I love to use them. They're really good for monitoring food intake, addiction, fear, memory, and so on and so forth. They just have all these complex behaviors that are really tractable for sort of human translation. And they also have this huge amount of tools available. So there's this system called DREAD. These are designer drugs where you can genetically encode a custom receptor and then feed in your own drug to activate neurons that you're interested in. And you can do the same thing with optogenetics, which I know all the students are aware of. And of course, there are these electrode stimulation experiments. You can do measurements of neural response prior to behavior or during. I don't know why I wrote that there.
I think your point here is there's lots of good reasons to use mice and rats.
Yeah, that is the overall point. I can't remember why this slide was here, but thank you, Kim, for that. But okay, actually, I do remember now. It transitions me into this little sort of precautionary tale that Kim's going to tell you all about. Even though mice are awesome, you need to really make sure. I told you before, they can't tell you if they're feeling or hearing things. There's ways to measure that Kim's going to tell you about, but you really want to avoid, if you can, behaviors that are sensitive to sensory artifacts or startle when possible. because Mikhail Shapiro, he sort of turned us onto this way of thinking. I mean, I think that ultrasound neuromodulation or stimulation is really sort of a hybrid of direct changing of cell activity through mechanical interaction. And then it's also, you know, a little bit of auditory and sensory sort of inputs, because the animals definitely can feel and sort of hear the ultrasound. And there's ways to bring that down, but you want to just be careful when you're monitoring this. So here I'm going to transition Kim in to make her case for whether or not I'm correct about this, and how
Yeah, I
she's
don't
been.
think about it this way. I think about it as the ultrasound stimulation is very different than the auditory stimulation, and that both of them can lead you to making measurements that you might misunderstand, but they're different. They are very different. But
Kim,
anyway, yeah,
why
so
don't
let
you
me
tell us
digress.
all about it?
Okay, so let's talk about what has now become the auditory confound. And this has a long history, actually, that knowing that ultrasound can be heard by animals. And so this is a piece of work that was done back in the 70s, where they showed they had an electrode in the ear of cats, I believe, and they were showing that when they applied ultrasound to the head, that you could get these oscillations here, and they call these cochlear microphonics. So it's been known for a very long time that animals could kind of quote hear the ultrasound, even though the fundamental frequency is way above the hearing range. There's something about the pulsing and the ultrasound pulse that makes them able to hear it. So what happened was more recently, there was a couple of pieces of work that were published in Neuron, and one of them was by Guo et al., where they were showing in hamsters, I believe, that they could measure a recording on their electrodes when they applied ultrasound, that then went away when they transected the auditory nerve. And so they're saying, well, clearly there isn't anything going on here other than this auditory effect. And then Sato with Mikhail Shapiro published a paper. Then these two were published at the same time in Neuron. And I'm quoting from the PubMed Central and Open Access pictures here. And what they showed was that they did some GCamp imaging, and they said, well, let's put the focus right down here in the visual cortex. And then they recorded the GCamp images, and what you see is, lo and behold, there's this auditory cortex lighting up on their GCamp images. So again, they said, okay, well, this is an auditory artifact. There's clearly something going on here, and we don't see very much here at the focus. So and then there's a concern, of course, that if you know, there's just a paper here that they referred to that was saying that if you have very loud sounds, then that can sort of inhibit other areas, and so that could potentially be what was going on. So in other words, this is very confusing. So we did some work to try to sort this out a little bit, and I have two things to show you. One of them was the work by Morteza Mohammed Javadi, where he looked at auditory brainstem response in, I've shown it here in a human head, but he actually did this in the mouse. And the idea is that once you stimulate with either airborne sound or ultrasound or whatever, if you stimulate the cochlea, then there's going to be the signal that comes in via these various nerves up into the brain, up into the auditory cortex. And that if you measure with an EEG electrode on the back of the head, then you can get a signal from all of these. And this is what it looks like over here. So this is a hearing wild type mouse recording. And this is just at one particular frequency, 16 kilohertz, that they're pretty sensitive to. And what happens is that you play the sound until, and this is sound thresholds right here. So you play the sound at increasing intensity, and initially you don't see anything. And as you increase the intensity, you actually start to see these oscillations. And when you see the oscillations for the very first time, you say, aha, that's where the threshold is for hearing that. And then at that frequency, you put a dot there, that's the threshold. And then you repeat this at different frequencies, and you come up with a hearing sensitivity. And so for the mouse, what we see is that they're very sensitive from about 2 kilohertz. We've plotted it at 4 kilohertz up to about 32 kilohertz. And then beyond that, it's just the threshold is way too high. And if the threshold is over like 80 dB SPL, then you kind of just give up and say, they don't really, they're not sensitive to it. So you can see that the mice are most sensitive in around 8 to 32 kilohertz. Okay.
Can you give us the sense of like how loud is 80 dB and how loud's 100?
So 40 dB is what I measured in the dining room when like the fish tank is on. It's pretty quiet to me, but when you get up to about 100, 120 dB, that's sort of jet engine.
Okay,
So
very,
yeah,
very loud.
that's pretty loud. Your sensitivity to sound is very nonlinear.
Mhm.
Okay, so he looked at 3 different types of mice. We had the wild type hearing mice. He had 2 different genetic knockouts, and how they were genetically knocked out, exactly what the knockout was, were slightly different. And so the fact that we got sort of the same result in both of those was sort of confirmatory. And in both of those, he just went through the process of testing their hearing to find out that yes, indeed, they were deaf. That you got up to 80 dB SPL, and you still couldn't measure these ABR measurements. So that meant that they were deaf. Okay, so he applied airborne sound with a speaker, and he applied ultrasound to their head, and then he had these EEG measurements so he could get the ABR, and he also had EMG electrodes in the forearms as well. Because at the time, we were mostly doing EMG measurements and then saying, how does the ultrasound acting on the brain result in a forelimb twitch? Okay, so this is what he got when he measured the ABR with the sound click, and you get these characteristic curves, just like we talked about before. This is a 40 dB SPL, not terribly loud. This is what it looked like when it's a sham. And then he applied ultrasound, and so he had 4 different waveforms. So down here is just a continuous wave that's a rectangle, and you see right here is the ABR immediately after that. So the time scale is a little bit different here. So these curves happen over about 5 milliseconds after the stimulus. And so we're seeing that right in here about 5 milliseconds after the onset. And then you'll also, interestingly, see it at the offset as well. And then if you pulse it, so we had 2 different pulsing schemes we show here. And if you pulse it, you sort of see the same thing, the onset and the offset. With this one, it was also a little bit interesting that you kind of see some activity throughout. And then he said, well, let's smooth out the waveform, because you could kind of think about this from a signal processing standpoint, where this has these edges right here have a spectral content that is very broad. And so if you smooth those out, then it narrows the spectral content. And then what you see is the ABR response diminished. And so that was his key finding, is that if you smooth the waveforms, then the ABR signal goes way, way down and is insignificant compared to sham. And at the same time, they still had a motor response. And so these were both the wild type as well as one of the genetic knockouts, and showing the success rate, like how often they twitched compared to how often you applied the ultrasound. Very high success rate for all of these different waveforms, including the smoothed waveform, and that was no different than the others. So that was saying to us that, yes, they were responding, and it wasn't due to the sound. We're not saying that they don't hear something. It's very clear they hear something, but the response wasn't due to the sound itself. So that was sort of our conclusion. And then we have a little bit of follow-up work in addition that I could maybe tell you about as well.
Love to hear it.
Okay. Yeah, there were lingering doubts, like what's going on here at the focus. So we wanted to go back and look at that in a little bit more detail, and then specifically with GCamp. So this is some work that Zhihai Qiu in my lab has been doing, where he put together a ring transducer. And the reason to do the ring transducer is that he can get a focal spot at the surface of the brain here, but also he could put his optics through the center in order to do the GCamp imaging. So that was his setup here. This just shows you the ring transducer and then the focal spot of the ring transducer with the hydrophone measurement. Okay, so this is a raw image. Then you put together the change in fluorescence, like Keith has already talked about, and you see something that looks like this. And then the focus is right at the center of the imaging field of view. So this tells you what his optics is able to see. And then this tells you here what's going bright is where the focused ultrasound was. Okay, so that's what the raw images looked like. And then he did this with square waves, and he did it with a smoothed wave. And I'll tell you a little bit about those. But first of all, I wanted to tell you just that he took those images, the change in fluorescence, and then he followed this paper here and looked at sort of the spatial content as well as the temporal content, and split it up into different components. And the reason then is that way he could identify, this is the raw image that incorporates everything, but then he could identify what's happening right at the focus based on the spatial and temporal dynamics. And then what about down here, which is where the auditory cortex is. So he could identify an auditory component, and then he could look at those two separately since he's separated them out. And then again, he looked at the square wave and the smooth wave. So this is what you see with the focus component with those two waveforms, and that you clearly see something at the focus, which looks very similar. I'll show you quantitation in just a moment with the smooth wave. So it's clearly having an effect at the focus. And then here in this one particular animal, which is my example that you see when you look at the auditory component, that there is an auditory component, but it's way, way diminished over the square wave. And then now looking at it just a little bit more quantitatively, just looking at those curves. So this is what's happening at the target and then at the auditory cortex. And just as a comparison in that Sato paper, what they found was that the auditory component was bigger and happened sooner than the visual component. And then what we find is that when we use the smoothed wave, that the target component is bigger and happens sooner than the auditory component. So the fact that we've smoothed it makes it diminishes the auditory component, as well as what you see there happens later. And so it's not causative if it happens later.
And it might be a good time to explain like your theories on why this difference is occurring. So why did you guys, see this sort of flip effect?
Well, I mean, I think it's just that with the smoothed waveform that there isn't the sort of the spectral leakage into the part of the frequency spectrum that they are sensitive to. So the smoothed waveform,
I think even with the square wave, you still get better target activation than they do, right? If I'm remembering correctly.
Well, I don't have in this presentation exactly what you see at the target, but you're sorry, with the square waveform. Yeah. So
I was trying to lead into, I think that you and Jorhai were speculating that angling the transducer can lead to larger propagation around the skull of shear waves, right?
Okay, yeah. So yeah, okay. There's a little bit of interpretation as to those differences. Yeah. So all right, let's come back to that in just a moment.
Okay.
Okay, so the other thing he did was he looked at the latency. So the latency means sort of the time to a certain point. So you could define that as maybe the time to the peak. What he did was he defined it as the time to 0.5% change in fluorescence. And what you can see there for those two different peaks was quite different. And so just sort of looking across a number of animals at the target, you see there's a lower latency than there is in the auditory cortex. It was a little hard to see in this plot over here, but now that he's quantitated it, it's quite a bit more obvious. And if you look now at a different paper by Mikhail Shapiro's group, where they looked at cultured cells with GCAMP, that they see a latency that's sort of similar to what we're seeing now, around 200 milliseconds. And that's about what we're seeing here at the target. So we think that it's with the smoothed waveform, our in vivo setup is more giving us a more direct measurement of what's going on. So there's two parts. The one part is underneath the focus, that it's a more direct measurement. But you do raise an interesting question, and I'd have to go back to the setup. And I'm not sure I have a picture of the two different setups, but ours is different in that the ultrasound is coming at a straight normal to the surface of the brain. And what they had done is that in order to get their optics to work, they didn't have that ring transducer. So instead, what they did was they had the ultrasound coming in from an angle, and then their optics were straight on. And when the ultrasound comes in at an angle, one of the things that we've talked about before in the class is the angle effect, that they're at an angle, you can have big reflections. And so one of the questions that is still open, because we haven't actually done the measurement to confirm how much of the ultrasound actually gets through the skull into the target when you have that sort of angle. And the reason why I say we haven't confirmed it, because we know this happens in human skull, but the mouse skull that we're looking at here is just a little bit different. Mouse skulls are very thin, they're less stiff than human skulls, but also this one, in both of these situations, they were thinned skulls, and then they had a little bit of dental cement. So it's not like a human skull, it's just a little bit different. They're still very thin so that you can get the optical signal through them. But we just have to go back and do that measurement if you're operating at an angle, how much ultrasound goes through and how much gets reflected. So that was one possibility here for what those differences were, is that the angle effect meant that there wasn't as much going in. We also was sort of hypostulating just to make up a word there, that that if you put the ultrasound at an angle, perhaps it creates more sort of surface waves on the bone, or the what is mimicking bone in our situation. And so that's something that we also really don't know the answer to. So I could have potentially have amplified the sound while diminishing the target just by having gone at an angle.
Yeah, correct me if I'm wrong, Kim, but the more reflection against an interface that you have, the more radiation force you may have at that interface. So the more like net displacement of the bone, right? Something like that.
Acting on the bone, yeah.
So Kim said if you have a higher angle like that other paper had, you're going to get more radiation force or sort of pushing of the bone, and that may lead to shear waves that propagate out and to the cochlea. So it all fits, and I really love your work, Kim. I think it's really it's awesome, as you know, because I've seen it many times.
Yeah, well, I have to thank Jerhai's done quite a bit of work here, so it's all his. Anyway, so I think that's sort of the summary here of what I have. Just that the smooth waveforms seem to be very effective. There are actually, some human work as well. I didn't have that in the presentation here, but there was some work that went on at that was presented at the Fun conference, and also there's a group at Oxford that looked at this as well. Just that humans can also hear the ultrasound, and then that's what the Oxford group did, and then the UCL. University College London presented some work where they had used a smooth waveform, and the humans said, yeah, they could hear it, but in many cases they heard less. So there's something to it, although that work wasn't sort of quantitated very, very much. So there's more to be done in humans. All right, and that's it. So I'm going to go back to you at this point, Keith.
Okay, so the last thing we want to talk about is using head-fixed animals, because a lot of these different methods, I mean, Kim showed you all of them, really. She showed you that she was using local field potential recordings from these auditory brainstem neurons. She also showed you GCamp from the skull. And it's worth noting that both of these studies were using head-fixed animals. And why would you want to head-fix the animal instead of having freely behaving? So we'll just talk about that briefly. So one reason is that you can use these really nice pieces of equipment that aren't necessarily mobile, like a large optical lens that allows you to look across a spatial field. The other reason is that the brain coordinates are all fixed in space. So you know that the animal is not going to suddenly turn its head and you're going to hit some different brain region that you didn't intend. And the last thing is important, and these tools are getting better, as I'll show you. Animals still can perform really interesting behaviors while constrained, and they can still be awake. So they don't necessarily have to be anesthetized or unconscious. So a little primer on stereotactic brain positioning. So what is stereotaxy? So stereotaxy is the idea that you have a predefined coordinate system of the brain. And we have these available for humans, mice, rabbits, all these different organisms. And the more genetically succinct your animal is with the map, the better prediction this coordinate system will be. So here we're looking at different brain regions in the mouse brain relative to landmarks on the skull. And this is the Allen Brain Atlas for mice. And to head fix them, you would put these little bars sort of in this little ear pocket, it's like right here, to stop the animal's head from rolling. So sort of like this and that. And then you would put a little sort of, not like a clamp, but a gentle, trying to make this sound nice, a gentle like piece in their mouth. And that stops the animal's head from changing pitch. So then if you affix the coordinates on the skull in the Z plane, where these two little skull marks called bregma and lambda create a plane, then you know that all other points in this brain atlas are relatively positioned to that plane.
Wait, so you're saying you can see the bumps for the through the skin?
Well, I'm glad you brought that up, Kim. Your questions are always nicely positioned. You have to pull the skin aside as I'm showing this gruesome image to the right. So you'll actually, snip the skin and pull it aside. And here you can see that these little sutures in the skull, where they all meet in the front, that's bregma. And then where these same sutures meet in the back, that's lambda. And again, when these create a plane, you'll then know the relative position as shown on the left of all these different brain regions. And then you can use dental cement to affix a sort of head bracket or a head holder to the skull. And the dental cement will typically seal all the vasculature in the skull in very nicely. So, the skull doesn't dehydrate or anything like that. So that's the way we do it. That's how we do it, and Kim does it, and all these different labs. So here you can see I'm going to compare. This is an experiment where I'm using Lfp recording. So here you can see these electrodes running wires into the brain. Here's my ultrasound emitting element, and here's a fiber to collect optical information. A lot going on here. But way on the left here, you can see actually a little head bracket. And here you can see the animal is kind of like, there's a lot going on the skull. His head is actually kind of strained from all the weight of these different components. You can still do it, but the alternative is to take this head bar that you can see here, these little, I'll actually just draw on the image. So here is the head bar. You can actually screw them into a bracket, which is shown here. And then underneath the animal, down here, you can put this styrofoam ball where we where we flow a little bit of air under the ball so it freely spins under the animal, and the animal can run on the ball as if it's sort of freely behaving. So you can see the animal's comfortable, and still all of our recording equipment is now unconstrained. And you can imagine we could add other ultrasound systems in on the side, or we could add a confocal lens here, as Kim does in many of her studies, fun things like that. And that's why head fixing an animal could be useful. And sorry, just quick on the screen here. Another way to allow the animal to move is to put like a ribbon or a treadmill underneath them. So here you can see I'm using like a standard like Christmas ribbon, even though it's not festive colors. It is a ribbon, and that will freely move under the animal so it can sort of run forwards or back up. Same principle at play here. There are more complicated, awesome things being made every day, and I won't go into detail, but here you can see the free-floating ball underneath the animal here, and sort of a confocal lens mount above. And what they're doing in this paper is they're presenting a screen in front of the animal. So here you can see a virtual reality room that the mice can run around in. And that allows you to present complex environments and to ask questions like, does the animal want to move towards this striped wall or this other patterned wall? And does it remember things? You can ask decision-making questions like that. It's very cool stuff.
So you can actually set it up so they're like kind of running through a maze or figuring out a maze.
Yeah, you can do a maze too. It's like the possibilities are sort of unlimited there. It's really, really neat.
But you can't really have like a maze where one end of it has like a treat and the other end does not have a treat. Because
okay, so here I'll pose the opposite. You can, if you put a little tube in front of the animal's mouth that dispenses sugar water. So people do what you're describing. And it's not like the treat's really in the maze, but if you dispense it when they go somewhere in virtual reality world, they think that they found a treat.
Yeah,
so it's sort of a cool abstraction of what you're proposing.
Yeah, pretty cute.
Yeah, it is cute. And here's a cool thing. So people argue that there's too much tension when it's fixed, but this group actually sensed rotational force. I forget how they did that, like using some sort of strain resistance measurement, and they would actually rotate the whole imaging apparatus to reduce that strain in real time using a closed loop algorithm. So even though the image is spinning, you can sort of register it to itself to get what you see on the right-hand side, a fixed image of these GCamp labeled cells being active. Again, this is just a way to reduce whatever this rotation would be. That would be I don't know, along the Z axis, just to reduce that strain for the animal.
It's just 1 degree of freedom with the rotation, so the registration seems pretty easy.
Yeah, and if you can notice, underneath the animal is a sort of floating disc. It's an air hockey table, so that the animal can push this thing underneath them with very little effort. You can see the little dots in the table flowing air onto this hockey puck. Mm-hmm. So let's put everything together. Here's a recording from Jordan Farrell of Yvonne Soltes's lab. So here he's head fixing the animal. He's using G-camp imaging and electrophysiology in the brain while the animal's on a floating ball, so he can measure the speed of the animal running. And I'm putting this here to illustrate that you can do all these fun different behaviors, recording methods, head fixation, all in like one experiment and collect massive amounts of data. And you can do this with ultrasound. So there's complex ways to merge all of these together. And here the animal's going through an epileptic event. So you can actually see spreading depression across the cortex. I think that was earlier in the video. Here, you can see activation just kind of passing a wave.
Wow, it's beautiful.
Yeah, it's a really cool video. Okay, so that's it.